letting me present this talk. It's really great for me. It's a pleasure for me. So I guess most of the audience, if not all of them, are familiar with the idea of in-cell NMR. So you can do a lot of really nice studies and characterization on proteins or other macromolecules in vitro. NMR is great because you can work in solution at physiological temperatures. So it's a great way to not destroy the sample and keep it in a close to physiological condition. However, sometimes you want to complement all the studies you can do in vitro and ask yourself what happens when I put this protein in the real cellular environment or in something which is as close as possible to a real physiologically relevant environment. That is why in-cell NMR was born 25 years ago or so. So the birth of the idea is there was the realization that you could somehow either deliver, introduce, or express a protein of interest inside an intact cells, and then run the NMR directly on the cells without breaking it. And it's possible, and it can be applied to do a lot of things. For example, if a protein can adopt different conformations and bind different ligands or metal ions, depending on the state of the cell or depending on the presence of other molecules or proteins, you can directly find out this. Or some proteins can have conformational variability. So depending on the physiological environment, they might adopt a different conformation from what you can find in vitro. And finally, the intrinsically disordered proteins could also adopt a different conformation and interact with partners. So it's a great tool to get biologically relevant information on the structure, the dynamics, and the interaction of proteins. Here is, I always like this picture from David Goodstone because it gives the idea. You have a lot of macromolecules inside the cell. Also, you have tons of small molecules, which are not depicted here, but you should not forget about them. And also you have metal ions. And in Florence, we have a long history of studying the interaction between proteins and metal ions. So you should not forget those as well. But about the macromolecules, what happens when you observe the behavior of a macromolecule inside this environment, you have several effects, which also affect the final, the readout, what you see in the NMR spectrum. One thing is macromolecular crowding, which is depends on the fact that this solution is really concentrated in proteins and nucleic acids, and therefore they prevent the diffusion of another macromolecule, such as the one that you want to look at. So you will have a slowdown in the translational and rotational diffusion because of that. Then there are all the possible interactions, and these are very dependent on the protein itself. So different proteins might behave in a completely different way. And these interactions could be attractive or repulsive. Think of electrostatics. And then you have different levels of interaction. You have strong specific interactions, which a given protein will have with its own partners. And then you might have a network of very weak interactions, which are sometimes called quinary structure or quinary interactions that can affect somehow also the behavior of the protein. Then you want to do this kind of experiments. You know that NMR has its own intrinsic limitations. It's not very sensitive, so it takes time to do it, and you always seek higher magnetic fields for some kind of experiments to increase the sensitivity. Now, if you want to do that with cells, you have additional challenges. And here's the summary. The low sensitivity of NMR is made worse by the fact that we cannot concentrate the protein like you would do in solution when you work with cells, because there is only so much protein that can be delivered or expressed in the cells, and also because you don't want to deviate from the physiological conditions. If you increase the protein concentration of one specific protein so much that it will be in large excess with respect to all the other components, it doesn't make sense anymore to study in that kind of environment. You have changed the environment itself. Therefore, there is a range which is also met by the fact that it's really hard sometimes to increase the protein concentration in a cell. And you don't want to do that. So then you're done with the concentration limits, which are even worse than what you can get in solution in vitro. Then we need to keep cells alive in order for these measurements to make sense. We could do in cell on NMR experiments on dying cells or dead cells, but then whatever outcome we get, it's hard to interpret because the cells were not in the correct conditions anymore. So we need a way to make sure that the cells don't die and they are possibly also metabolically stable in the instrument. And I will show you some ways to overcome this challenge and achieve this. Then on top of that, there is transverse relaxation. So the effect for which you get severe line broadening of the peaks in the NMR spectrum to such an extent sometimes that you cannot even see the signals anymore. So this is also what happens in vitro when you have a pure protein. And you probably all know that it's very dependent on the protein size and therefore on the molecular weight of the macromolecule. Then in cells, because of the attractive interactions with other proteins, it can happen that even small proteins become invisible when you do some kind of NMR spectra, which are so nice when you can get data from them. So that's also a limitation, very protein dependent. So some proteins could behave better than others. And in this case, up to some extent, we can help this by overcome this by using tailored NMR experiments, exploiting, for example, the Trosi effect or CRENEPT. But then sometimes the broadening is so much that we have to switch to different kind of nuclei, switch to methyl groups and side chains, for example, instead of the backbone, or switch to totally different kind of spectroscopy. And I will show some examples on that. Then moving on, I will briefly show how different ways to prepare cell samples for NMR. I'll start from the oldest one, and it's still very useful whenever you want to study the behavior of a protein in a bacterium. You can use bacteria E. coli cells. So Escherichia coli is the workhorse for every NMR lab where you want to make purify recombinantly express and purify labeled proteins. So they are easily adapted to in-cell NMR. The only difference is that you don't purify the protein. You make the cells express the protein, provide isotope labeling, and then put the cells in the NMR tube. That's exactly how it's done. So we usually start with transformation of bacteria and then grow the biomass of bacteria, of transformed bacteria in unlabeled medium. Then once you have enough cells, you induce the protein expression. And at the same time, switch to a labeled medium. And bacteria can also grow in minimal media. So this is also not expensive in the best cases. And then after protein expression, which hopefully takes place over a short time, the labeling will be selective enough because the cells did not grow so much, but the protein expression took place in labeled medium. At this point, it's a matter of collecting the cells and resuspending them in an appropriate buffer and then put it in an NMR tube for NMR. Then if you want to move to eukaryotic cells, there are a few alternative approaches. The one I will show at first are conceptually different from the previous one because here proteins are delivered to cells. That means that you first purify the protein from recombinant expression and purification. You have total freedom of isotope labeling. And then the protein somehow has to be delivered to the cells in which you want to study it. And this was shown for the first time by Gerhard Wagner and Phil Selenko, that it could be done on frog oocytes, Xenopus levis. These oocytes are quite large, so they can be micro-injected. It takes something like 300 oocytes in a Shigemi tube to fill the active region and be able to shim the magnet and collect good NMR spectra. And This is very convenient because they have a huge cytoplasm, so it's very good to study cytoplasmic proteins. And as long as the protein of interest can be concentrated to very high concentrations, because we cannot inject more than a few picoliters or nanoliters, sorry. So to reach a decent levels and concentration in the final sample, it needs to be concentrated. And then there is the microscopic equivalent of the injection, which is translocation mediated by peptides, cell-penetrating peptides. One can make a fusion construct where a cell-penetrating peptide, for example, from HIV TAT protein is fused to the target protein, and this will induce, then once monolayer mammalian cells are treated with this protein, the CPP tag will induce fusion through the plasma membrane, and the protein will be translocated inside the cytoplasm. A similar way, but it's kind of, alternative way to deliver proteins inside the mammalian tissue cultures is by using a toxin, streptolizin O. This will make pores on the plasma membrane. Indeed, this is part of the category of pore-forming toxin. So as it makes pores, these pores are large enough that small proteins can diffuse through the pore and reach the cytoplasm. Then, of course, also the cell content will leak out. So this procedure has to be carried out by carefully. So one has to calibrate the incubation time in order not to kill the cells. The good thing is that by treating the cell culture with calcium ions, the pores are sealed, because this triggers a very big conformational change on the toxin, so that it will close the pores. And at that point, the ideal would be to do cell sorting in order to select only the cells which took up enough protein for NMR, because some cells will not be permealized, and therefore they will not contain the labeled protein. Then finally, kind of a more versatile tool is electroporation. In this way, you can still make holes in the plasma membrane. There is no toxin, there is an electric field, pulsed electric field. So by pulsing a cell suspension with a very strong electric field, one can hyperpolarize one side of the plasma membrane, and this will make holes electrically, in fact, through which the protein can diffuse. Then once the pulse program, the electric pulses are shut off, then the plasma membrane will fill again. And by then selecting the cells which did not die, one can make them recover on a plate and then recover the live cells, and they will contain the protein. So this was shown to be applicable to different cell lines, which is very good if you want to study protein, which is relevant in a given type of cell. Then I will now focus on an approach which I contributed to develop some time ago in Florence, which is the protein overexpression in human cells. Just like I showed you in bacteria, human cells can also be used for overexpressing proteins. I say overexpressing because we still need a quantity of protein which is usually larger than the physiological amount. But that is a limitation of NMR. If you want to do NMR, you need to increase the protein level. This can be done by exploiting constitutive expression, vectors for constitutive expression in mammalian cells. HEC293T cells are especially good for protein expression. They are also used for purifying membrane proteins, for example. And we can employ uniformly labeled media. These media are more expensive than what you can use in bacteria because they are chemically defined, but they contain all or most of the amino acids, and all of them are isotope labeled. So these media are more expensive. However, the costs are partially spread because they are cut by the fact that we don't need a very large volume of medium. I will show you a few numbers now. This method is quite versatile because once you have set up a good combination of vector and transfection method and cell line, then changing the DNA, changing the gene, does not lead to huge differences. So it's very good, for example, if you have a wild type protein and then you want to screen a series of mutants, the protocol is basically the same. And if the protein is expressed, then you can then run a lot of transfections. That means delivering the DNA to the mammalian cells and make many samples. So that is the very basic description of the protocol. We first grow the hex cells as a monolayer. Then when they are about 50–60% of confluency on the plate, transient transfection is performed. We need a transfecting reagent here, and we are using a very cheap one, which is polyethylenamine, which can be toxic for some cell lines, but not for hex cells. So the cells will be totally fine. I see a raised hand, so I think I can also answer questions as they come, but maybe the moderators can tell me if I should answer everything at the end, or we can do it.
Yeah, we can wait till the end, I think.
Okay, then I'll wait to the end. Get to the end. So after transient transfection, all the cells will take up many copies of DNA, so that will ensure expression along all the cell population. And the protein expression takes place over 2 days, in which after 2 days we reach a plateau. And then it can go on for longer, but then over time the plasmid will be lost because it's not integrated in the cell genome. That is why it's called transient transfection. So after 2 days of protein expression in an isotope labeled medium, if needed, then the cells are collected for NMR. This is the vector that we use most of the time. It was developed in Oxford for expression of proteins, of secreted proteins. Therefore, the original version has a secretion sequence, a secretion signal just downstream of the start codon. But if this is removed, then the protein expression takes place in the cytoplasm. So this is an excellent way to overexpress proteins in the cytoplasm. It's constitutively active, so there is no need to induce expression. As long as it gets into the cells, the expression will take place. And this is an example. This is not a Western blot, it's an FDS page stained with coumasin, and like you do with bacteria. And you can see the protein bands for most of them. That tells you that the protein expression can be very high. And it's also true that it's dependent on the specific protein. But we have successful expression for many different, wildly different proteins. So I would say this is quite a robust approach. How do we then put these cells in the NMR? There are many ways. I will show you the easiest one, perhaps. We want to increase the density of spins in the tube as much as possible, right? Because of sensitivity and because if you double the number of spins, then it takes a quarter of the time to get the same signal to noise. That's how NMR works. So you want to really push that to the limit, but you cannot concentrate the protein inside each cell. You can make a pellet of cells in order for them to occupy as much volume as possible in the NMR tube. And this is actually easy because these cells do not stay afloat. So by gently spinning a tube, we can make a pellet inside the tube. These are a few numbers just for reference. We can grow hex cells at confluence in a 75 square centimeter flask, which requires 20 milliliters of medium. Hence, each sample is not hugely expensive because these volumes are quite low. And this will, for hex cells, will correspond to about 20 to 30 million cells. Now, these will, once they are spun in the NMR tube, they will make a soft pellet which covers perfectly the active region in a 3 millimeter Shigemi tube. We could increase the signal by using a bigger tube, like 4 or 5 millimeter Shigemi, and then scale the number of cells accordingly. However, in my experience, at very high fields, magnetic fields, the ionic strength coming from a thicker sample will negatively affect the signal intensity. So in the end, it takes it's cheaper, takes less pain in the lab to prepare less cells, and you can get basically the same amount of signal in a 3 millimeter tube. Then, which kind of concentration we're talking about? So the thickest band of the gel I showed you before corresponded to about 150 to 100 micromolar concentration inside the tube, which means that in the cell it's higher than that. That is clearly not physiological. However, some proteins are natively expressed at quite high levels in the cell, so that means in a relative sense we are increasing the levels of protein, but not too much, not by too many orders of magnitude. Then some of the lowest expressing cells were a few tens of micromolar. And nice thing is that if you think you are expressing too much protein, we can tune down the expression levels by mixing DNA with non-coding DNA, with an empty vector, so that the transfection procedure will still work in the same way, but every cell will take up less copies of encoding DNA. And this is shown here. We can just dilute the encoding DNA, and the protein expression levels will go down. So it can be controlled to some extent. The detection limit depends really on the type of protein. It's highly protein dependent. Intrinsically disordered proteins, when they don't interact with the environment, can be detected as low as few micromolar concentration. There are works by Tilson-Ekman and Francois Tellier where they show that they can really reach 10, 20 micromolar in the cell. Then folded proteins, if they interact and stick around, they are just undetectable. Whereas if they don't, then you need a few tens of micromolar, maybe 100 micromolar if they're big. So it depends. And the detection time for these kind of pellets is advisable to stay below 2 hours of acquisition. But then depending on the cell density, if you make a suspension or a pellet, that might change. However, we usually run 1 hour experiments on 3 millimeter Shigemi tubes and keep the cells at 37 degrees and at high fields. So this is the mileage we get in these conditions. But again, that can change. Then an important aspect is make sure that the cells did not die during the experiment. This is easily done by using staining, such as trypan blue negative stain. This stain will be uptaken by the cells which are damaged, which are dying. So it's easy. One can just treat the cells with this stain, and then the dead cells will be stained blue, whereas the live cells will be not colored. So by counting the total cells and then the blue cells, we can estimate the percentage of dead cells. And then by comparing them at the beginning and at the end, we can assess how much the cells died during the experiment. We usually try to stay above 90% of live cells in order to be able to interpret the data. So finally, then, this is an example of a proton nitrogen so fast HMQC spectrum recorded over 1 hour at 950 megahertz on a folded protein, which is superoxide dismutase 1. I will show some application on this protein. Do you notice something? Yes, you do. In the middle, there is this blob. This does not arise from the protein, whereas the other cross peaks clearly belong to a folded protein. What is in the middle is caused by the non-specific incorporation of nitrogen-15 during protein expression. So this nitrogen-15 goes everywhere in the cell. And so we will get a background. The nice thing is that HEK cells, human cells, are very reproducible. They always behave in the same way. So one can repeat the experiment by transfecting the cells with a non-coding DNA. What we then get is just the background signal. And it can be algebraically subtracted very easily in topspin. That can be done in topspin. And then you can recover the signals which were hidden down here. So in the end, it can be applied to many unfolded and folded protein, this approach, and we can get very good or decent NMR spectra. I will show, and then these were uniform labeled, but we can perform amino acid selective labeling. And these are two examples. One is with methionine with carbon 13 on the terminal methyl group, and then also the backbone can be labeled with nitrogen 15. And this can be done just by making your own custom medium or buying a custom medium lacking one amino acid and then providing the labeled amino acid. So nice thing about human cells is that they require most of the amino acids. The essential amino acids will not undergo scrambling. So they will be incorporated in the right positions in the cell, in the protein sequence. So it's quite easy to selectively label some types of amino acids in this expression system. This is the first application of this approach. We could monitor the maturation of copper zinc superoxide dismutase in human cells. I will make a long story short, but you can appreciate how the spectra change as you treat the cells differently. So in the first example, in this spectrum, you see a protein which did not bind any metal ion, because once it's overexpressed in the cells, there is just not enough zinc and not enough copper to allow for total complete protein maturation. This is monitored both by uniform labeling and selective labeling on cystate. Then if the experiment is repeated in the presence of zinc and copper, I'm skipping some steps, but we went step by step. So we could see the zinc binding, then we could see the zinc and copper binding. In order to do this, we also had to co-express a chaperone, a metallochaperone for sod. But in doing so, we could reach the final mature state of the protein. Then the cysteine chemical shifts also revealed the formation of an intramolecular disulfide bond, which is a native bond which is necessary for the protein to be structurally stabilized. Now, this was also applied to a panel of disease-related mutants of this protein. They are related, they are connected to the onset of myotrophic lateral sclerosis. And it's important to understand how they affect the protein maturation. So we could also apply this approach to a series of mutants and see how the maturation was impaired, and then it was recovered by the co-expression of the chaperone. By monitoring both the proton signals, here's just 1D proton NMR in the immunoregion, and this is by doing selective labeling with cystates. A few more words about selective labeling. We have been recently working on ways to improve the readability of the NMR spectra, and one aspect is spectral crowding. Sometimes you have too many signals, and for intrinsically disordered proteins, for example, this is even worse because these signals will be all piled up in the center of the spectrum. So we need a way to make these spectra simpler and be able to interpret them. As I said, we can label single amino acid, but sometimes you want to switch to side chain labeling, for example, methyl labeling. In bacteria, one can use precursors. But what about mammalian cells? So they usually need ready L amino acids. So what we found recently, together with Robert Conrad and Roman Lichtenegger, is that we can actually provide precursors also in human cells, alpha keto acids. Thanks to the activity of human transaminases, which are reversible, catalyze the reversible reaction, we can provide the alpha keto acid and get an amino acid of choice. And this was shown, so instead of providing a labeled amino acid, we can provide a labeled precursor, and the cells will convert it to labeled amino acid. This was shown for tyrosine, phenylalanine, and these are the side chain C13 labeled peaks from an HMQC, from carbon HMQC, and also methyl groups of leucine and valine. So it's quite versatile. And then deuteration further helps narrowing the line shapes, the line width. Then finally, we can also introduce fluorinated amino acids. We showed that aromatic amino acid can be introduced. Here, the expression phase needs, it's a bit different, because if we provide fluorinated amino acids, which are chemically different from the native amino acids, the cells will not be happy. And at the same time of transfection, which is also stressing the cells a little bit, providing fluorinated amino acids in place of the native ones will result in poor viability and no protein expression. A solution to this problem was to switch the medium after a recovery phase, after transfection. So first, the cells expressing proteins will be not without fluorine, and then switching the medium after, like 8 hours, and then providing a medium with a fluorinated amino acid will work. And these are the aromatic fluorinated amino acids which were tested. We even purified the protein from these. So this is carbonic anhydrase containing 7 tryptophan residues, and all 7 tryptophan residues were shown to have fluorine incorporated. So the incorporation works, even though it's not 100% incorporation. And we can do fluorine NMR. Now, bear with me. These were recorded on a very old probe, which was not made for fluorine. So it's a miracle we could even get signals at all. Nowadays, we have a QCI, quad-channel cryoprobe, with 2 independent proton and fluorine channels, and also a newer cryoprobe with proton that can be tuned to fluorine. So with the right instrument, you can get much, much better spectra than this, or the same spectra in a fraction of the time. But as you can see, most proteins gave nicely resolved peaks, and some of these proteins I will not explain too much because of time, but these proteins were not detectable by nitrogen HSQC because of line broadening. But when you do simple fluorine 1D spectra, you can still get broad signals, but you can see the protein. So I think it's extending the size limitation or tumbling limitation of in-cell NMR. Now, what about time limitation? I told you that after 2 hours or more, these cells will start to die if you make a pellet in the tube. So that's not good. If we need more time to measure things, we need to find an alternative way. And the solution to this is use a flow bioreactor. That is a device which is able to keep the cells alive for longer times in the NMR spectrometer by providing a continuous flow of nutrients. This has been done in many different flavors by so many groups in the past. So I will only show a few more recent examples where the objective of the study was to actually observe protein signals inside the cell. So there is, for example, a bioreactor, a batch bioreactor, a batch flow bioreactor by Gary Pilak group, where bacteria were encapsulated in beads, and then the flow was coming from the bottom, where the cells would mix with fresh media and then settle back down in the active coil. Then there are more recent examples of Shigemi tubes or just normal 5mm tubes. One from Taka Ito and Ichiro Shimada group with the Shigemi, and then from Alex Sheckman group with standard tubes. In both cases, nutrients are applied through a pump or a syringe and through the cells, and the cells are encapsulated in spaghetti made of gel, which can be a special gel which melts if you cool it down and makes a gel if you heat it up, such as mobile, or in low melting agarose, which does not dissolve back if you put it at 37 degrees. So following these great examples, we also made our own flow bioreactor in Florence by using a flow unit which can be bought. So it's commercially available, and then also applying the cell encapsulation protocol in low melting agarose. So the cells are placed inside the flow unit of this device, which is then assembled and allows the flow of nutrients inside. Everything is kept at a controlled temperature, so it can be kept, for example, at 37 degrees. These devices then put in place of a normal tube inside the magnet, and the pump, I've tried several types of pump. Nowadays I'm using multi-channel pumps, and I will show later what you can do with different channels and different reservoirs. That's an example. So we can preserve cell viability to very high levels, up to 3 days of running this bioreactor and running NMR experiments. The cell viability in the presence of fresh nutrients was higher than 90%. And ATP production was also happening. So the cells were alive and sufficiently stable for and metabolically active. This is a picture of the cross-section of this agarose spaghetti with the cells inside. And then with a few minutes left, I will show more recent applications. I've been focusing more and more on drug design, drug development. I think this is a in cell NMR have the great promises for these applications, because we want to make new drugs. We want them to be very active towards the target, but we also want to show that they actually bind the target inside the cell. And we also want the drugs to be able to reach the target inside the cells. So it's not enough to just optimize the molecules in vitro and then check that they bind very strongly the target in vitro. And it's also not sufficient to just do cellular-based assays where you find out that the cells stop growing or they die because you treated them with an anti-cancer drug if you don't even know that the drug is actually binding the target that you wanted. So I think in cell NMR, it's good within the drug development pipeline. This is a very old slide because it's a very old concept. We start from in silico screening of millions of compounds. Then we start doing fragment screening in vitro. By the way, NMR is great for fragment screening. And then build new molecules, lead compounds, optimize the lead compounds by changing functional group and find the best ones. And then we start treating the cells with them. At that point, we don't even know if they are really targeting the right molecule. So somewhere here, when you start doing in vitro assays on cell cultures, in cell NMR could fill this gap. And we showed this a few years ago by taking a very nice protein, which is carbonic anhydrase. It behaves very nicely when you do NMR. So you can actually do heteronuclear NMR and you will get great high quality spectra. It's an ubiquitous protein. There are 16 isoforms in humans, and some are related to cancer. So it's also a relevant pharmacological target. Here's the zoom of the binding site, active site, where the inhibitors bind. To bind, they have to displace this water molecule here, and this will inactivate the protein. Nice thing is that we can not just look at the backbone of the protein by NMR, but we can also look at these proteins here. They are very close to the ligand binding site, and therefore are excellent probes for target engagement. Therefore, there is the binding of the ligand to the target. These are the signals from these immunoprotons. They are not immunoprotons technically, they come from imidazole, but they resonate in the immunoregion of the spectrum, which is very clean. So even on intact cells, you can actually look at these signals when you overexpress the protein. And so we tested a few ligands and show that some would actually pose a shift in these signals and telling us that it would bind the target in cells, whereas for many others, nothing happened. So after an initial questioning of everything, then we found out that these ligands were not able to cross the membrane and reach the target. So we proved that there was no target engagement. On those who did reach the target, we could show that there was a dose dependence and a time dependence of the amount of bound target in the cells. And this is because it takes time for these drugs to penetrate the cells. That means that we could measure the diffusion through the plasma membrane. And this is, long story short, we could find the correlation between the predicted permeability in silico and the absence of binding in cells. And we could also see a correlation between how fast these two molecules could bind the target, could cross the membrane, and how much drug we need to take for treatment of glaucoma. So the drug which permeates faster is actually recommended at a very much lower dosage for treating glaucoma. So there is a correlation with potency. And that proves that I think it's useful to do this kind of measurement. Then I want to measure more quantitative things. For example, can we assess the binding affinity in cells? And I think we can by exploiting competition binding. So the principle is quite simple. If you displace a strong ligand with another strong ligand, you can measure a displacement curve and be able to determine the ratio of the affinity constants, the dissociation constants of the two ligands. And these, by measuring the fraction of protein bound to one of the two ligands, we can obtain these, we can use this simple equation, and with the due approximations, of course, but they are correct in the specific setting I will show you later, and ignore the free protein because these ligands are strong enough, so they will saturate the protein. And so with the simple equation, we can get this ratio by measuring the fraction of protein bound to each ligand. I showed this. We should this by labeling the carbonic anhydrase. This is selective labeling with histidines. And then by using the multi-channel pump with the bioreactor, by changing the concentration of one of the two ligands, whereas one was kept at constant concentration, we could reproduce a displacement curve. So this is time, and step by step, a test ligand was delivered to the cells at increasing concentrations. So that's the, again, scheme of the bioreactor. Two channels with two reservoirs. Both contain a reference ligand or SPI, and only one contains the second ligand. And then by changing the flow rate, we can decide the final concentration of the second ligand. This is what we get from the NMR. Step by step, the first ligand is displaced, and one can see the appearance of the second set of peaks coming from the second ligand bound to the target. These are the pure protein with bound ligand 1, ligand 2. And these are the displacement curve. Then by fitting the plateau values of these displacement curves, we can fit these values with the formula and get the ratio. And then assuming we know the dissociation of one, we can calculate the dissociation constant of the other. But very often you just need to rank compounds and find which one is the most potent in vivo. And so you don't need absolute numbers, you just want the ratio. And this is measured by the experiment. Then in 1 minute, I'll show that we can then switch to ligand-based NMR whenever it's impossible to observe directly the target, such as the case of the N-terminal domain of HSP90, which is a different pharmacological target. This is invisible in itself because of interactions. So because of spin relaxation, we cannot do HSQC in cells and see this protein. But what we can do is then use a fluorinated ligand and use it as a SPI to see how much a second ligand is displacing the SPI, and then do the exact same measurement from a ligand point of view. So here are two peaks. One comes from the free ligand outside of the cells, and one comes from the ligand bound to the target inside the cells. And then same principle. We then provide step by step a second ligand, which will displace the first one. Here is the displacement curve. Here is the fitted curve. And again, by knowing the KD of one, we can derive the KD of the other, or better, just keep the ratio. It works even better if both ligands are fluorinated. This way, you can be fully quantitative. You can trust that even if the total amount of target changes over time, you don't care because you know the total by integrating both peaks. So hopefully this works. Here is the bioreactor run over 60 hours, I think. So you can see the displacement of the SPI, which is on the right, and the increase of signal from the test ligand on the left. And then by integrating everything, we can then just fit globally and obtain the ratio of KDs. And with this, I will finish. This is our current group, which I of course thank, and then also the collaborations, which of the works I showed in this seminar. And I thank you all for your attention.
Thank you so much, Professor Luchinat, for the fantastic tutorial about how to do in-cell NMR in different ways and their applications. So yeah, let's open the floor for questions. Already see that there is some. So first question, does it have to be with 900 or 950 megahertz? I think this was about the mammalian in-cell NMR work.
No, that's a very good question. I mean, in principle, whatever instrument can give you sufficient signal-to-noise ratio is fine. So as with the NMR in vitro, by the way. So no, for example, I've been using a cryoprobe at 600 megahertz and recorded very nice, high-quality in-cell HMQC spectra on a globular protein. I think it was carbonic anhydrase. So clearly, you may have slightly lower spectral resolution and sensitivity because of energy level splitting and so on and so forth. But No, I think any high field instrument equipped with a cryoprobe will be sufficient. And then sometimes the relaxation that you have, the spin relaxation, gets worse when you get to very high fields. So sometimes higher is not better, I would say. For example, Flora NMR was recorded at 600 megahertz, either with a room temperature probe or with a cryoprobe. And I would not advise going to very high fields for Flora NMR because fluorine is affected by a very, very large chemical shielding anisotropy, which will dramatically increase the transverse relaxation. So you actually want to keep lower fields for Flora NMR.
Okay, cool. Well, we have two questions here from Sarah. So thanks a lot for the great talk. One very practical question. Okay, so to the selective labeling in mammalian cells, how much precursor or amino acid do you need compared to E. coli cells?
Much precursor. Tough question because, to be honest, I never did that in E. coli. So I'm pretty sure we need less. So what we did is to provide a 5-fold or 3-fold excess with respect to the amount of L-amino acid which is present in the normal mammalian cell medium, which is the MEM in our case. So we took that value and multiplied by, we did a few tests, 2x, 3x, 5x, and 5x performs a little bit better because you increase the amount of amino acid which is produced by the cells from the keto acid precursor, but still the total volume is limited to 20 milliliters in our case, which is less than what you usually do when you grow 1 liter of culture of bacteria. So it's clearly two different scales. We make a cell sample which we don't use to purify protein. So clearly the protein production, the yield will be different, will be much lower. So it's not so straightforward to compare. But for the specific purpose of doing in-cell NMR, I think we can do with less precursor with respect to making a preparation of protein in bacteria and then purify that protein, then of course you can do a lot of things with that. So it doesn't apply. It's hard to compare, I would say.
Mm-hmm. Yeah, so another question to the fluorine NMR, I think you have published on that. Would it be advantageous to use aromatic amino acids that harbor fluorine C13 spin system to exploit the TROSY effect for nicer 2D spectra, or is it the effort of synthesizing these amino acids is just too much?
Yeah, also a very great question. And indeed, yes. We showed again with Roman Lichtenegger and Robert Conrad that who provided the fluorinated amino acid labeled with carbon 13 on the carbon bound to the fluorine 19. And yes, definitely, yes, it's possible to exploit the CF TROSY effect. I honestly didn't go on with that yet, but I think that's also a promising thing to do. It's a promising approach because it, in principle, can allow, it overcomes again the slow tumbling of proteins, of folded proteins inside the cell. So it should be possible to perform in cell on things which would otherwise be invisible because of spin relaxation. Having said that, I cannot comment too much on the synthesis. It depends on the kind of amino acids and the synthetic route. So it's all a matter of finding a smart route to make a fluorinated carbon 13 labeled amino acid, which is not hugely expensive. But so I would be on the other end of the pipeline. I'm the user. I'm the one who would gladly buy a cheap C13F amino acid, but I cannot comment on how crazy is it to synthesize it.
Mm-hmm. Yeah, so what a task can be solved using F19 labeled protein, the fluorine? we just see several signals
Yeah.
in the 1D.
True, yes, yes, and they were poorly resolved. So that was, in a way, a proof of principle. You can introduce fluorinated amino acids in proteins. However, the incorporation will take place in all positions. So the more you have in the protein sequence, the worse it will be to separate them in the spectrum. That's true. Tryptophan is uncommon in protein sequences, so you might have one or two tryptophans. So it becomes easier to interpret the spectra. Then carbonic anhydrase was a special case because it has 7 tryptophans, and so you start to have some overlap. So we could show that ligand binding makes some of these signals shift, so it can be used directly to probe ligand binding, just like I showed with N15 label and with ligand-based NMR. But probably the best use of protein fluorination will be coupled to carbon-13 and TROZ, because that way you can separate the signals in 2D spectra. It's easier to interpret than 1D spectra.
Okay, well, nice question. Yeah, I was wondering about this too. I guess it's for selective, to see how ligand is selective. So you throw different ligands, and when you have just fluorine, and also yeah for the TROZ. So I have a question, but I'm gonna just go through all the questions. In terms of the fluorine NMR, have you tried any NOE? Fluorine proton experiment.
Proton fluorine NOE or Halsey. Yeah, no, I didn't. I'm aware that this is a quite insensitive experiment because the transfer is not very efficient. So the short answer is no, I didn't try, but might be possible, yes. Especially maybe when looking at the ligand instead of the protein, then maybe the relaxation properties might be more favorable. But that's a speculation, I don't know.
Okay, cool. What's the cutoff point for how much protein to overexpress? What would you say is the lowest degree of overexpression you could get away with while still getting decent data?
Tough question.
Okay.
I would say for physiological relevance, the lowest the better. So if that is paramount, make sure the protein does not go beyond certain levels, then I would switch to a long acquisition with a bioreactor and try to keep the cells alive for as long as possible and accumulate signal with a low expression level. So what is the lowest degree? I would say for well-behaving proteins, 10 micromolar intracellular concentration would be a limit. However, with a bioreactor, in principle, one could even reach lower expression levels. So again, it strongly depends on the setup and on the specific protein. But in the best case for line shape, that is intrinsically disordered proteins, and in the best case for acquisition time, that is with the bioreactor, maybe we could reach a few micromolar. There is a very recent paper published in JACS by François Tellier, where they do this kind of reasoning. Although they did not use a bioreactor, they just put the cells in the instrument. But I think they calculated somehow this based on the expected improvement in signal to noise ratio for an IDP with a given acquisition time.
So a follow-up from me on that, actually.
I cannot hear you.
Oh, I said I follow-up question.
Maybe you can speak closer to the mic. I don't know. I have trouble hearing you.
Can you hear me now?
Very
No?
weakly, low volume.
Oh, okay. Well, now?
It's a bit better,
No?
yes.
Okay, well, actually, I'll just Yeah, I'll just go ask the. I have
Now,
a follow-up
now I can
question.
hear you. Sorry, yeah, now I can hear you.
Oh, okay.
Perfect.
Okay, so I have a follow-up on that actually, because you
Mm-hmm.
said if you worry about making so much like overexpressed protein, then you dilute it with the empty vector, right? So
Yes.
why would you worry about having overexpressed? Is it like for toxicity, or like why would you
That
worry about
depends.
that?
Yeah, no, that's a fair point. It depends on the protein. I mean, in the case of globular soluble proteins, which are in the cytoplasm and catalyze very fast reactions such as carbonic anhydrase or superoxide dismutase, these proteins are natively expressed to quite high levels, tens of micromolar in a cell, which is exceptionally high. So in that case, I don't worry of increasing the expression levels by tenfold, for example. I would say it's acceptable. It's an acceptable ratio. But then if you had a protein which can really be toxic to the cells or can trigger some pathways, if it's overexpressed to high levels, then that would be an issue. And that's why it's important to know that you can tune down the protein expression if needed. I hope I answered the question.
Oh, yeah, perfect. Okay, we have several people asking as well about shimming. How difficult is it
Huh.
with this
Yeah,
heterogeneous sampling?
it's surprisingly easy in my hands, at least, as long as there is no air or gas bubbles of any kind in the tube. Shimming will just work. With the normal top shim routine, or even by manual shimming, we did that on the old probe for Florence NMR spectra. They are shimmed just like normal tubes. What will not be shimmed is the microscopic inhomogeneity, which comes from the fact that you have cells and not a clear solution. So you have a microscopic level inhomogeneity. That's unavoidable. But the macroscopic inhomogeneity of the field can be fixed just by shimming normally.
Okay, perfect. Following question is much scope for use of Q? Oh no, sorry. Yeah, about the CSA of the fluorine. I was wondering what would happen if you measuring it directly
Hmm.
in the cells.
Oh, just the same as it happens in vitro. I mean, the transverse relaxation of fluorine depends on the dipolar relaxation term and on the CSA term and the CSA scale. They both increase as you increase the correlation, the rotational correlation time. So the bigger the protein, the slower it tumbles, the higher broadening you have. And CSA has a quadratic dependence on the magnetic field. That means that while dipolar relaxation will basically be constant, more or less, when you are at high fields, it will not change too much. CSA will make it worse with the square of the field. So the higher the field, the worse is this term. Now, for proton NMR, that's not a big issue. For carbon, sometimes it is, with some carbons. For fluorine, it blows over the roof. Like the CSA term for fluorine, especially for aromatic fluorine, is really big. So when you increase the field, you will see the difference. And cells or in vitro, it doesn't matter too much. You have an additional contribution of broadening due to the fact that you are in the cells. But the CSA is a big issue, and that will also be in vitro.
Cool. Fantastic. Well, we have here a question about the scope of use of QNMR, particularly for phosphorus related to ADP, ATP, to not just confirm viability, to quantify growth state or status of growth, plateau state, etc. Could internal or external standard be an option?
I guess so. I didn't focus too much on that, but in principle, yes. I mean, assuming one can find not much of an external channel, maybe an internal reference signal that tells you how many cells you have, and then check whether the ATP, ADP signals change over time. They stay constant, they increase, they decrease. That can be used to quantify the metabolic activity of the cells. And then In principle, I guess the answer is yes, although I cannot really tell myself.
Okay. Yeah, the second question about the Shigemi tubes versus the standard 3 millimeter. Yeah. Do you think that you get comparable?
Sorry, now I cannot hear
Yeah.
you anymore.
So do you think that you get comparable results can be achieved with this one versus Shigemi versus the standard 3 millimeter?
I expect basically the same results. We use the Shigemi just so we don't need to fill the tube, the bottom of the tube with cells, considering that the coil cannot actually see the content of the tube down to the bottom. So that content is a waste. You have to fill that part of the tube, but you get no signal out of it. So switching to a Shigemi allows you to do the same with less cells. But if you have you have plenty of cells, you can fill a standard 3 millimeter and you should get exactly the same amount of signal.
Okay, perfect. Well, deuterium labeling, is it helpful in cell NMR?
I think definitely yes. In all cases where dipolar relaxation, proton dipolar relaxation makes leads to line broadening, such as the case with slow tumbling proteins. Whenever you look at protons from those proteins, they are broadened because of dipolar relaxation. So deuteration would help definitely. It's however not so easy to achieve uniform deuteration in human cells because they cannot grow in deuterated media, unlike bacteria. So one approach would be to actually provide a series of, to do selective labeling of side chains of, for example, aliphatic residues, providing a lot of deuterated aliphatic residues at the same time. That should decrease the proton density inside the hydrophobic core. And then if one of these contains a CH3 group and the carbon is labeled with C13, that should lead to improved relaxation properties.
Cool. So I also probably missed this part. How exactly
Can't hear you
are...
anymore. Sorry.
Okay.
But if
Sorry. Yeah, you can read the question,
I can,
which is about...
okay. Sorry.
Yeah.
Let me open the Q&A. Maybe it's easier. Okay. How exactly are the protein of interest are labeled specifically in cell? Because you mentioned the cells are growing in labeled media, so everything has to be labeled. Good point. Excellent point. The labeling selectivity is not very good when you do protein expression in human cells because the protein expression phase is very long. It takes 2 days. Yes, also other proteins will be labeled in the cells. So that's always an issue to be aware of when we do protein expression directly inside the cells. The labeling selectivity will not be great, whereas the other approaches I showed at the beginning, where you deliver proteins, will provide much better labeling selectivity because the cells are not labeled, whereas your protein is labeled. So yes, that's a trade-off. I really like the protein expression approach. It's very versatile. We could do a lot of nice things, but the labeling selectivity will not be great. And sometimes you can do background subtraction. Sometimes you can do a control experiment and skip and don't use it to do a subtraction, but just an overlay, and then check whether some of the signals should be disregarded because they are coming from other proteins. So it's a sort of negative control. That's doable as well. So I'll move to the next. So I can also see who is asking. Nice. So Jesus Anglo, wonderful talk, Enrico. Thank you. Do you think it would be possible to detect in-cell interactions with tens, hundreds, micromolar KD ligands? Do they have to be nanomolar? Jesus, we should talk. No, I mean, I want to find that out. So you read my mind. That's something I'm working on right now, and I still don't know the answer, but that would be awesome if we could somehow measure low affinity ligands. in cells. So hopefully I will have an answer soon, and hopefully it will be yes, but I don't have it right now. And then final question from Sophia. Hi, Sophia. We are going to study the effect of mutations on protein ligand binding. The ligand natively present in the cell and the protein is synthetic. Protein is unstable and can be purified. And we try to solve this task by in-cell NMR. So if you have proof of protein expression in the cell, but it's just too unstable to be purified, maybe yes. I mean, nice thing of in-cell NMR with the protein expression approach is that you don't need to purify the protein. If it's possible to do everything in cells without having to rely on in vitro data to interpret the in-cell NMR spectra, and that is an important point, then yes, you could, in principle, if the protein behavior is such that you can detect signals, or you just care about the ligand binding, you can do ligand detected NMR, then in principle, yes.
Okay, cool. Perfect. So answer. No, can you hear me?
Yes, I can.
Okay. Well, I have last question from Julie, from the one of the panelists. She was saying, yeah, what's the next big step in cell NMR that you are most excited about? And we're gonna end with that question.
Yes, I'm really going forward to use this tool to probe protein drug interactions in the cells. So the next step there would be to find a way to perform these kind of experiments, either by looking at the protein or by looking at the ligand with improved cell models. That's a big challenge, and I didn't mention anything in the seminar, but there's a lot of effort also to improve the preparation of the sample in terms of which kind of cells and how uniform the protein expression are by both François Tellier and Lukács Trentírek and myself. And so I think the next step will be being able to move to more complex cell models, such as the 3D cell cultures, steroids. This was already demonstrated, but possibly also from different cell lines with different phenotypes. That would be a big improvement, both to investigate proteins inside the cells and protein ligand interactions, because that can be affected by the phenotype of the cell. And in terms of protein observation, structural characterization, I would go for IDPs, intrinsically disordered proteins, because I think it's more natural to apply NMR to such proteins because they cannot be studied at the single residue level with any other technique other than NMR. And yes, so I think these are the two aspects that I look more forward into.